Yep, started screen sharing. There we go. We see you are a banana enthusiast.
Yes, I am. It's really something I enjoy, and essentially I can really talk hours about this topic. But I try to focus this mostly on my work during my PhD at Wageningen University and what I'm doing currently, especially focus on two important things like the diagnostics of the tropical race 4, but also the breeding part. So uh, as you know, my name is Fernando. I'm from Colombia. I I did my PhD at Wageningen University, and actually I started to work in this topic from 2012, and I've been dedicated exclusively to this, to bananas and Panama disease since then. Before of that, I was a coffee breeder, so I am very happy that I'm working in two of my favorite crops, both with similar problems. Currently, I'm a postdoc working at Kijin. It's a company of technology that helps breeders, among other things, to go faster with the process. So when we talk about bananas, as we discussed at the beginning, the first thing that comes to our minds is this, right? The perfect yellow banana that you can find in every supermarket around the world. That is essentially considered like a sweet banana and used for desserts and also for other, it's considered in a different way. But it's important to know that, especially in the US and here in Europe, when we talk about bananas, people think about just one because we don't have more. So lately, it's been incorporated a little bit more the cooking bananas, but essentially what we can find is Cavendish bananas. And actually, Cavendish is not a variety; it's a group of varieties or cultivars or clones, whatever you want to call it. And it doesn't matter which sticker you see in the banana, essentially is the same. And there are many different types of cultivars like Grand Name, Willis Pollo, Valerie, but all of these are essentially Cavendish and are clones. They differ from each other just for very small things like size, color of the stem, these kind of characteristics. But for the rest of the world, especially Africa and Latin America, bananas are more than the Cavendish. So we have the cooking banana that is actually a staple food in many countries, and it is actually more important than the Cavendish banana. It's used in many cultures. Every part of the banana is used, the stem, the leaves, everything, and even in some countries, you can make alcoholic drinks with beer. The banana diversity is huge, so we know one, but if you go to other places, especially Southeast Asia, you will find a lot of more diversity in the market. They are not as pretty as the Cavendish, and that's one of the reasons why the industry especially says that we don't have more bananas maybe to consume. But as you can see in this slide, there are more than thousands varieties, including triploids that are the pretty ones, diploid cultivars that are local cultivars, and when I talk about hybrids, is products of breeding, as well as many other wild accessions. This is just a taste of the diversity of banana. These are diploids. I always say for people to understand that these are not pretty. For me, they are, but they are not as we expect. You see, they are like not ready for the market. So they are cool, red, different taste, but you cannot bring them or export it to other places because of many other factors. These are the commercial bananas. You see the difference: productivity, shape, and the most important, they are seedless, because the previous ones, they still produce some seeds. The center of origin of the banana, but also the center of origin of the pathogen, is Southeast Asia. This is just a taste again of the wild relatives of the banana, the original bananas. As you can see, different shapes, but these ones, we cannot eat them because they are just full of seeds. There is nothing left that you can eat. But these are important because these ones are different to the triploid material like the Cavendish. These ones, you can use them for breeding. because they are fertile, you can cross them, and they are more or less, let's say, compatible to each other. The challenge is to transfer those traits to an edible commercial banana. This is not really important. Is the most important of this slide is to know that there are different, that almost every banana, edible banana, belongs to the Musa general, and there are several sections. Nowadays, we are talking about only 2 sections because they mix this and they reduce the number to 4 to 2 only, based on the number of chromosomes. The most important of this slide is to know that most edible bananas are coming from these 2 ancestors, the Musa acuminata and the Musa balbisiana. And that's why sometimes you can hear people talking about genome E on the bananas and also the genome B. Those 2 are the ancestors of every banana that you can eat. This one, the Musa acuminata, provides the genome A, and the Musa balbisiana provides the genome B. Interspecific crosses between these 2 and a lot of years give us what we have now. So a lot of selection and domestication, especially. So there are reports that says that the domestication of the banana started 7,000 years ago. So that's what it cost to get what we have now, the bananas without seeds, because in the past they were all seedless bananas. But through process of selection, we have what is the parthenocarpic bananas that we have at the moment. You know that bananas is a big business. It's very important crop. Actually, is the most important if we talk about fruits. But if we want to understand why bananas are under risk now, we have to really go and travel in time. Before the Cavendish, there was another banana that was the Gros Michel, the Big Mike. This was a fantastic, I said was, but actually it's still alive. It's not extinct, as the media says that it was extinct because of the Panama disease. No, that's not true. We still produce a lot of Gros Michel in Latin America, but we cannot produce in big amounts because of the Panama disease. And I'm going to that in a minute. So it's important to mention that the Gros Michel was, people say that it's superior, but if you really see it, it's essentially more or less the same as Cavendish. But the difference with the Gros Michel is that the Gros Michel is a very rustic material. It's very strong and it has a very thick skin, which allows to move the bananas like this. So this was the way how people move bananas before. So they just cut the plants from the tree and drop them in the ships and send them to other places. So the first report of the Gros Michel is in 1830. And from there, it started to disseminate the Gros Michel, the banana, from Martinique to the Caribbean and beyond. So everything was completely covered with Gros Michel. And the first losses of the reported for the Panama disease on Gros Michel were in 1890. So a few years later, there were a lot of problems with a disease that started to kill bananas and that make people think that was really going for extinction. And the responsible at that time was reported as the Panama disease, race 1. The pathogen is known as Fusarium oxysporum forma special cubense, race 1. Is the one that caused the problem in Latin America. So we are going in detail of this, but this is just to show you now, to give you a sneak peek of how it looks in the lab and in the field, externally and internally. So the 20th century was very important for many people. I like to talk about this, and also difficult for many others. And in this period of time, there were things that really changed the life, in this case, the history of many. And I like to say, if you allow me to say it like this, that also there were one event that changed the history of the banana, because when the first big problem with Panama disease started to really affect the Gros Michel, that really changed everything. in the industry, because that was the only banana that was cultivated. And at some point, there were not more bananas. And that was caused by one of the biggest epidemics in the history of the agriculture, that was caused by the fungus in the tropical, in the Gros Michel. So that was not like in one day everything changed. No, it was gradually, but in this period of time, that around 30 to 50 years, it was not possible to do this. This was a dream again. It was not possible to produce a Gros Michel. Of course, there was clever people that tried to save the banana at that time with different techniques. Like this is one, for example. They floated the areas, and actually that works for some time, because as you mentioned at the beginning of our conversation, that kills everything that is in the water. So kills good things, but also kills bad things that are in the soil. But this also, as you know, not everything dies, and this also contributed to the dissemination of the pathogen to other areas. So after doing this technique that helped a lot during the 50s, they were able to plant bananas again, but this was not for a long time. After the 1950, this was really a dream, this part. But there was hope, and that was the discovery of the Cavendish clones. These were resistant to that specific race of the pathogen, the race 1. And we can say that the Cavendish, like a miracle, saved the industry. And it's actually really like a miracle, because there is not at least I cannot think, I don't know, you guys maybe can think in another example, a great example like the banana, because this Cavendish is being resistant to the race 1 for more than 100 years. That's not happening in other crops. You know that the resistance is broken after a few years, but in the banana, with the Fusarium, race 1, these Cavendish are still resistant. The same soils that were used to grow the Gros Michel are now used to grow the Cavendish, and nothing happens with the race 1. So this cultivar really contains is genetically great. But what was the problem? Again, people made the same mistake, growing areas like oceans of bananas, Cavendish type, thinking that that would never happen again. But unfortunately, and starting in the 90s, people noticed that Cavendish plantations show it now the symptoms of the Panama disease. So what happened here, people say? Again, there was considered like the Panama disease version 2. It's a different race, it's genetically different, and actually it's a different species, but it's still considered in the group of Fusarium oxysporum. So it was called Tropical Race 4. It affects Cavendish bananas, but not only Cavendish, and that's the biggest problem, because it also is able to kill other genotypes, including the cooking bananas that I mentioned earlier. And it's something also very important, that why we call the pandemic now, because when we discovered it for the first time outside of this area, because this was the first area, it was a local problem in Southeast Asia and not a Northern Australia. But then from local problem, it became a global pandemic, because in just 5 years, we passed from 6 countries with the presence of the tropical race 4 to more than 20 at the moment. So we discovered it last year in Colombia, and this year, unfortunately, is also in the Mayotte Islands. So it's moving fast. Probably it was there for already long time, but there were not tools to identify the fungus. Oh, they existed, but they were not as easy as we can do it now. So at this time, I would like to make this short break here, not break, this bracket, just to talk about that. We had been working back in the university when I was at the PhD there. I was in charge, together with my colleagues, of the diagnostics and evaluation of samples from around the world. So we received samples from all over the world to check different types of symptoms, and it's very important that we also did it from soil, from these parts of the banana, and the soil is something important and something difficult to control. So everything that you observe here in red were positive for TR4. The ones in white were negative. So this allows to report the disease in different countries. So as you can see here, the first report of the Panama disease tropical race 4 affecting Cavendish was in 2000. Actually, this was in 2013, but you know that the publications came later. Later, we found it in Pakistan, Lebanon, later in other countries, but also in this part in Vietnam, Laos, and finally last year, unfortunately, in Colombia. So of course, other researchers have also found it in different places, and that's why we say that now we have more than 20 countries infected with that. It's important also to mention that the techniques to detect the tropical risk 4 have had a lot of development in the last years, in the last 10 years, because before we had only the very rustic techniques like by eye in the field, which is very important. And then it was this technique that is called the VCGs that you might not, and I will explain later. And of course, DNA-based techniques are now the ones that we use to detect the potential presence of the tropical race 4. And of course, we pass from months to just minutes now. And even at Buckingham University, we develop a pipeline that we call the forensics that allows to more or less estimate the potential relationship between strains collected in different areas. In this case, for example, you can see that the Jordan and Lebanon are very similar, and they are not that similar to the Vietnam and Laos, but the China and Myanmar are also similar, and Philippines, Pakistan. So this is just a new technique that is fast to detect. Also, indirectly, we have worked in the diagnostics of other projects. And this is also very important because here I would like to really say something that maybe people don't like to hear, but it's very important to say that the tropical race 4 is very, very dangerous for the bananas. These researchers in Australia, they have these plants, the transgenic resistant plants, they have them in Darwin, and they have to send the samples to Queensland for the analysis. But they prefer to send the samples to the Netherlands rather than send them in free areas of Fusarium inside their own country. I'm saying this because I hear that there are projects in Latin America where they are thinking to do tests in the infected areas, for example, in Colombia. So they want to do, for research purposes, experiments in those areas, while other people are trying to eradicate. There are plans to increase the inoculum in the soil for research purposes, which is not, in my opinion, is not. It should not be like that, because if the Australians that have the highest level of security send the samples to other places to prevent movement of material between their own country, just imagine what can happen in Latin America. So these are other papers. It's not important. We have been giving workshops in Latin America. And by the way, if you want to download this document, it's free. All the protocols for diagnostics are, for now, are in Spanish, but because we did it for the Latin American people, but we are working on the English version, but you can download it on my ResearchGate or in the Comunidad Andina page. So it's a complete book that has all the protocols, but also a nice introduction about the Panama disease. So this is the pathogen, and this fantastic picture shows the hyphas of the pathogen just coming out from through the stomata of the banana. So here you can see it inside of the plant. Let me move this. And this big picture that you see in the middle is a magnification of one of these things here on top of this picture. As you can see, the Fusarium is colonizing everything, the empty spaces, but also the bundles, the vascular tissue of the banana plant, but not only the empty spaces, also crossing cells, as you can see here. So it doesn't have a cure. When the plant is infected, we know that it's going to die. So Fusarium is a big group of fungus. As you know, there are more than 100 species. One of the most important taxons in the agricultural world is the Fusarium oxysporum complex. They are endophytes, saprophytes, but also pathogens, not only in plants, also in humans. They cause wilting on plants, and there are more than 100 forma speciales. These forma speciales are specific to specific names that the taxonomists give to the fungus to really show that they are very, how do you say that? They are just specific to certain hosts. That's why we have here the Fusarium oxysporum forma special PC that is specific to peas. And we have the Fusarium oxysporum lycopersicum, for example, that affects only tomatoes. And we have the Fusarium oxysporum cubense, that is the one that affects banana. So by the way, do you know why it's called cubense and not, for example, Fusarium musa or Fusarium banana or something? Actually, it's because the first samples that were analyzed by Dr. Erwin Smith in 1910, they were received from Cuba. So he decided to call it Fusarium cubense. To go to the Fusarium cubense forma special, oxysporum forma special cubense, is a complete story that you can check if you explore this hashtag, the history of the banana. So the Fusarium of the banana causes the disease that is called Fusarium wilt or Panama disease. Belongs to the Fusarium oxysporum forma special cubense. However, my colleague at Bacchini University revised this taxonomy, and she proposed a new name because based on the genetic markers, she found out that the Fusarium tropical race 4 is actually a different species. So she decided to propose the name Fusarium oxysporum, Fusarium odoratissimo for the tropical race 4. There are 4 races. The Fusarium produces this kind of conidia, so micro, macro conidia, and chlamydospores. These chlamydospores are very dangerous because, according to some researchers, they can survive in the soil for even decades. So the soil can be contaminated for decades. And how do they know? Because they put Gros Michel in areas where there were not bananas. There were not bananas for more than 30 years. And when they put the Gros Michel there, those bananas were infested. And so far, there is not a control, not even chemical control, and it spreads very easy. Soil, water, plants. So the life cycle is very basic. Essentially, the spores that I showed you previously enter in contact with the roots. They cross the cortex and the cells of the roots, and then they reach the tissue, the vascular tissue, where they colonize, especially the xylem, causing this reddish discoloration inside and blocking the movement of nutrients and water, and that's why the plants get a wilt, and then later on they die. So here we have 3 races of the Fusarium affecting 3 different cultivars of the Fusarium, and actually 3 different groups even. So here we see a Fusarium race 1 affecting apple banana, which is genome AAB. It means that it has 2 genomes of the Acuminata and 1 of the Balbisiana. Here is race 2 affecting plantain or cooking banana, genome ABB. And here we have the Cavendish, AAA, tropical race 4. If you see, there is not really a big difference in the symptoms, and that's why it's very dangerous, because the tropical race 4 can be affecting other cultivars and just be there, and people can think, okay, this is just race 1 because it's in the Gros Michel. And that's one of the biggest problems, and that's what we think happened in many countries, especially in Colombia, that people were just thinking that was another disease, because these symptoms are very easy to observe with many other things, not only biotic, but also abiotic factors. This is a very typical symptom of the Fusarium. It's called the pseudostem splitting. So internally, the pseudostem of the banana just explodes, as you can see here. It's not present in every plant, but this is a complementary symptom. Internally, all this reddish discoloration on the vessels shows the presence of the Fusarium. So normally you don't see these red lines, it's just white. You can see here all these. So for the diagnostics, we take one of these pieces and we continue with all the process of re-isolation. The races, as I mentioned before, race 1, race 2, and race 4, and the race 4 divided in two, in race 4 tropical, race 4, and race subtropical, race 4, depending on the area where they affect. But the problem that we are facing now is with the tropical race 4. They are also classified, a part of races, in 24 VCGs, group compatibility, vegetative compatibility groups. So this is a technique that's been used for many years to really identify unknown Fusarium in this group of Fusarium cubens, Fusarium cisperum cubens. So this is just a scheme of how the Fusarium of the banana is classified by races, but compatibility groups, and of course using molecular markers, DNA. So the most important here is that you remember that the tropical race 4 is just one. It's a clonal strain and belongs only to just one VCG, while the other ones, like for example the subtropical, that also affects Cavendish, but only under certain circumstances, like for example when the plant is stressed. So here, this is just one, and this is very ambiguous and very weird, this classification, because it's not really accurate, and it's a very old technique, and that's why we like to work with sequences now, with sequencing. As you can see, some researchers catalog the subtropical race 4 in the VCG 12.0, and others in 12, and others as race 1 because they isolated from Gros Michel. So, and this is not the only example. This one, for example, subtropical. Why? Because they isolated from a Cavendish. That's why they call it subtropical race 4. But also, in some cases, has been isolated from Gros Michel, and that's why they call it race 1. So that's why it's better to go with molecular markers. I know that in some countries it's difficult to go for sequencing, but every time it's been more accessible. Everything you see here is all the diversity, the known diversity of the Fusarium that exists at the moment. So the 24 VCGs. All these are the Fusarium that are able to affect banana in general. Doesn't matter if it's Cavendish or everything. But the most important is to recognize that the VCG 1213, this is actually one. It was just classified for two independent groups at the same time, but later on they realized that it's just one. So that's why it's called VCG 1213-16. It's just one. So this is tropical race 4, tropical race 4, and everything in orange is the race 1. And sometimes, as I mentioned before, it's also subtropical. Here we have something that we always call the clone wars, because we have in one side a clonal variety, and in the other side we have a clone that is the tropical race 4. So my colleague at Wageningen University also collected the tropical race 4 for many places. She made all the analysis, phylogenetic analysis, and she came to the conclusion that everything related to Cubense tropical race 4 is just the same. They differ in a very little, in a few SNPs, but everything is the same. It's just a clone. The impact is, of course, huge and depends of where, because in some countries, like for example, Australia, Colombia, Philippines, the impact is bigger because they are producing countries for the export. In other places, it's mostly for local market, which is also very bad because many people depend of the cultivation of bananas. But it's not as huge as Philippines, Colombia, and Ecuador, for example. As you can see here, this is just an oasis of bananas in Jordan, from where the samples were collected for the diagnostics. And this is in Lebanon and Pakistan, as you can see here, is all devastation with the tropical race 4, because the problem with the tropical race 4 is that if you find an infected plant, it's not enough to remove that plant. You have to remove thousands of plants around it and close that area, and that area you cannot use it again. Some people say we can grow beans, we can grow rice. Yeah, you could, but you have the risk that you have to go to that area and also bring the Fusarium on your shoes, on water, on the tools. And that's what happened in the greater Mekong, for example. All this area that is borders with each other, we came to the conclusion that the Cavendish was actually the vehicle of dissemination of the tropical race 4. Of course, also together with the limited awareness and the lack of quarantine measurements, but the Cavendish being the vehicle of dissemination of the tropical race 4. In this graph, you can observe the number of reports of Fusarium tropical race 4 starting in 1977 to 2020. So now maybe you're asking why is that old? Because I said that it was around the beginning of the 90s, right? It's because actually the tropical race 4 existed, of course, but it was restricted to other areas. And it was really noticed when the big plantations of Cavendish started to be affected. But the first report of the tropical race 4 was in the 70s, as you can see here. But then from 2012, when we discovered for the first time outside of Southeast Asia, look at the number of countries that have been affected. This was published yesterday, actually, or the previous days. This is in Australia, which I call a good example, because as you can see here, the number of, we are talking about plants. And every time there is a new plant, they try to destroy the area. So I think they founded 85 plants, and it's like 5 farms since the beginning of the problem, only in Australia, because they have really the best measurements to contain the disease. And if we compare with China, for example, this data, we are not talking here about number of plants, we are talking about hectares. As you can see, we passed from 60,000 hectares to 100,000 hectares from 2014 to 2017. This is the Colombia case. I was very sad, let's say, because I had to confirm that the Panama disease was in that area. So I traveled there last year, actually, during this time, to collect the samples and to analyze them. And I had to confirm the presence of the pathogen in Colombia for the first time in the whole continent. And that was the biggest fear all this time, because in Latin America, we have the biggest banana producers, right? Ecuador, Costa Rica, Colombia. If you go to supermarket, if you haven't noticed that and you go to the supermarket and you check the sticker, for sure you will see in the sticker Colombia, Ecuador, Panama, Costa Rica. So every banana you eat comes from this area, and that's why it's that bad that the pathogen arrived. Colombia has been doing a great work with the OPPN there to maintain the disease there in those areas, but also to destroy. As you can see here, as I mentioned before, it's not that you have to eliminate one plant and cross around and that's it. No, here we have to really eliminate a lot of plants. As you can see in these pictures, and I like this quote because it shows how severe it is. So this number of plants has been destroyed, and currently 1,700 out of this number of hectares are under quarantine. What does it mean? That people cannot enter. So it's just closed farms, and it's a big number in that area. So almost the 60% of all the cultivated area in that specific area, because it's in Colombia, we have 2 main areas of banana production. This is one of them, it's the small one, but this is in the Guajira area. But you can see all these farms, they all are in quarantine, these 7 farms. Of course, all the measures have been established to maintain the disease there. But I also say always that we found it dead there in June. But that doesn't mean that the pathogen arrived in June. So the pathogen, to show these symptoms, being optimistic, it needs from 6 to 1 year, right? So we need to be careful there and continue with all the diagnostics. and try to really destroy and eradicate, not only plant, but try to also to eradicate the fungus from the soil. There are some ideas. So what have we learned from the Gros Michel and the race one? Really not much, because essentially the same thing is happening again, right? So the history repeats itself. There is not awareness, even though in some countries, because of the new arrival of the pathogen to Latin America, Ecuador, and all these areas have really enforced all the rules. And if you go to the airport, for example, you really have to spend some time there to explain where you come, where if you have been in contact, is like the COVID-19 now. It's really, have you been in contact with the bananas? Have you been in contact with in areas with tropical race 4? And so I was already kind of familiar with the situation that we have now with the COVID, and that's why we compare it with the coronavirus. Because before we traveled to different places, it was the same always. Like, have you been in contact with the tropical race 4? Have you been in farm with tropical race 4? All this, and so there is quite similarities. So the soil is contaminated. People just move to other place or change the crops. They lose income. There is not a replacement. There are many people working, and I'm going to cover that in the coming slides, working in different approaches to bring new bananas, and there is not a control. So here we have the strategies for control of the tropical race 4. So many people are working on these different approaches, and essentially we have two scenarios. One is countries without the tropical race 4 and countries with the tropical race 4. For countries without the tropical race 4, this is the key, the quarantine and the exclusion. So we don't want Fusarium here, put everything that is necessary to keep it outside. But if you already have a tropical race 4, you cannot control, but you can manage it. So there are many people working in the biological control, chemical control, that actually these two especially, they work very well in the lab, in the greenhouse. But when you want to transfer these technologies to the field, it's just impossible, because actually the tropical race 4, the Fusarium, is very sensitive to everything. You can kill it with bleach, but how to make that the product enter in contact with the pathogen? My colleague Maricar Salasinas from the Philippines, she established a depth of the Fusarium, and she was looking at which depth in the soil is present, and she found that even 1 meter down. So there is not a product that can go 1 meter to kill the pathogen. So these two work very well in the field. There is new technologies that the university and other researchers are establishing to try to make it work in the field. The cultural control is what all the activities that the farmers do in the farm to protect the plants. This is just one of the methodologies used in the Philippines. So when they find an infected plant, they cover it with this rice hull, and then they burned this for 1 or 2 days, thinking that the heat of these flames will kill the Fusarium. That works only for the very superficial parts of the soil, but not for deeper areas. And of course, as you can see here, people are without boots, without just walking there in this plant. And the most risky way to transfer Fusarium from one place to another is the shoes, because if the pathogen is in the soil, you can bring it. Myself, I have re-isolated Fusarium from soil from the shoes. So we are focused essentially in the genetic control. And if we talk about the genetic control, we are talking about breeding. And what are the options for breeding bananas? You know that bananas are triploids, are sterile, are difficult to breed, but it's not impossible. So what are the options? We have pre-existing genotypes. So what is that? What we already have in the field. As I mentioned before, there are thousands of bananas, but we need to see if those are resistant, and we need to educate the industry and also the consumers to get used to new things, because not all the bananas taste the same. As you can imagine, there is this apple banana, for example, that really tasted like apple. So people should be more open to try new things. I don't know what your opinion about that, but I like to try new things. So I don't know what's your opinion. So we have also conventional breeding, that is use of crosses, and the non-conventional breeding. We are talking here about transgenics, mutagenesis, and gene editing. So what do we need for the breeding? Doesn't matter which technique, we need genes, right? We need resistance. And that's what I did during my PhD. So I entered in contact with all these institutions around the world. They have collections of bananas. They have more than 1,500 banana accessions. So we did a pre-selection of 350, and I managed to evaluate 245. So it can look like a few, like you didn't do much, but it's very difficult. This is not Arabidopsis, this is not tomato. Growing bananas is difficult, and you have to test big bananas to see this. So this is the total of plants that I evaluated, but also I have to improve the protocols for phenotyping to evaluate if one plant is resistant or not. So here you can see this nice example. This is one banana that's called Zebrina, because it looks like a zebra. And these ones here that look like a pretty ornamental yellow bananas are not really happy. They are going to die very soon. These two are infected bananas, and this one is a healthy banana. So because I had so many plants, sometimes more than 1,000, so I had to develop new protocols for the evaluation of such a number of plants. So this is published and it's available for everyone. I also made some videos on how to do it. Those are on my YouTube, so you can share later if you want and you can see. But this is the initial step. Once we have resistant potential parentals, we can go for either of the options for breeding. We are working on breeding. In the company I'm working now, it's called the Crop Innovation Center, KeyGene. And we have 40 accessions that we selected for resistance, but also not only for resistance, also for color, shape, and other characteristics, because we really want to incorporate new diversity. And we have, of course, the Cavendish, because one of the main focus is to develop a commercial banana very soon, but after that, continue with the different diversity. So I did all the pre-breeding work during my PhD. I evaluated many hybrids, wild types, and cultivars, and I'm using those ones now for the crosses. So I'm doing crosses, but also we are doing something that's called more precision breeding or molecular breeding. It's not just taking this and cross. No, we already sequenced. This is more than 100 cultivars, and these ones with the asterisk here are materials that are resistant. Unfortunately, are not ready, are not edible in some cases because are full of seeds. But we have already the sequence, and we can really make groups with this. And our approach, because there are two kinds of breeding: the pedigree breeding, that is crossing this with this to create a new plant, and that one cross it with another one to produce a new banana. But it's more complex than this, because as you know, these are triploid bananas. Our approach is called reconstructive breeding, and that means that we want to really recreate a Cavendish banana. That's one of the objectives. How? We are not going to cross a triploid with a diploid to create tetraploids. What we want to do is really make a new Cavendish, and for that we have to go to the ancestors. So something interesting is that the Gros Michel and the Cavendish are actually, we can call it cousins. They are very close. They have different parentals, but they have the same mother. We can call it like that. Our idea is to, why not choose one of the resistant ones to try to recreate Cavendish banana. So having all the genetic information give us a little bit more space to select and not lose a lot of years crossing things that we don't know if they are going to work. So I'm 2 years already as a postdoc, and I already developed the F1. So these bananas that you observe here with seeds, I already phenotyped them. They are resistant to tropical race 4 and to also race 1, because the race 1 is a prerequisite, right? You cannot put a resistant banana to tropical race 4 but susceptible to race 1. So this is resistant to both. And now the challenge is to transfer those genes to an edible banana. So we have to transfer the genes, but also remove all those seeds and make it bigger. But of course, there are strategies, double haploids, that we can also try to incorporate as another different techniques to develop new bananas. Also talking about traditional breeding, recently we started also collaborating with ITA project, Breeding Better Bananas in South Africa. It's also another group that is working to develop bananas for the African market. Those are tetraploid bananas that are used a lot in these areas. So we are also part of this nice project. Talking about non-conventional breeding, we are not doing that. So I'm not working with transgenics. Probably we will do it at some point to validate genes or something, but we are really focused on traditional breeding accelerated with technology. But we collaborate with James Dale, that is one of the most important people working in transgenic bananas. And actually, he developed a resistant banana that is transgenic, and actually it's a cisgenic because the genes that were collected that were inserted in the Cavendish came from another banana, from one of those diploid cultivars that are resistant. Also, people started to work in gene editing, but of course, for gene editing, you also need genes, and so far, unfortunately, for the banana, it sounds really weird because it's such an important crop, and for more than 70 years, there was not knowledge about genes, and even for the race 1, nobody knows why the Cavendish is resistant to the race 1. So there is not research in banana. It started 10 years ago, more or less, but before that, there was a gap of research on banana, mostly because people, or that's one of the hypotheses, that people focus mostly to investigate in the other big problem, that is the black sigatoka in banana. So because that's really a problem, but it can be solved by applying a lot of pesticides. So another technique that is used to develop new bananas is the mutation. We also worked with people in Israel with this company, Rahan Mary Stems. We evaluated thousands of mutants, and actually we found some that were less susceptible, and those ones are being investigated further by this company in Southeast Asia. Probably they will have success to bring resistant or less susceptible bananas sooner. So for us, at least for me as a person, the most important is for the banana world, I think, is the diversity. So if we have hundreds of apples in the shelves, why not more bananas? So that's my dream for the future, and I hope we can see that. Just as take-home messages, I would like you to remember that the current pandemic of the Panama disease is caused by one single clone, the tropical race 4. We need standardized protocols to phenotype, in that case, that we can compare from one lab to another lab. Not everything is lost because the resistance exists. As I show in the big screening that we perform, we identify more or less 13% of the accessions that are resistant. So most things are susceptible, including not only Cavendish plantains, cooking bananas, but there is resistance, and that we can probably use. The control measurements are really poor, and Fusarium will continue spreading. So we have to be alert that there will be more incursions of the pathogen, because after Colombia, that was last year, this year it was reported in Turkey and the Islas Mayotte. And another thing is that one of my colleagues also identified different strains of Fusarium in Indonesia that can really infect banana, but that are not outside yet. So they are specifically in some areas of Indonesia that at some point can just escape. So the risk is imminent there. And with this, I will finish. Thanks for your attention, and I will be happy to answer your questions.